Welcome, students, to Module 4, where we begin to focus on evolution in terms of its mechanisms. And, this term evolution kind of has been thrown around a little bit in the class, but hasn't really been clearly defined. So, in this segment, what we're going to do is we're going to unpack what evolution is, what it isn't, and what is a modern synthesis. How do we interpret evolution and a number of different mechanisms that, right, are, what is actually causing change in populations. So we can observe the change, and then, that's kind of observing evolution, recording it, and then the mechanisms are, what's actually causing that change. We can put ourselves in the modern synthesis because, or also, it's also known as neo-Darwinism. That's what every evolutionary biology class that's being taught all over the world has to contend with the modern synthesis, and it oftentimes is explicitly two components: Darwin's ideas about natural selection, and then Mendel's experiments from pea plants that led him to discover at least one type of inheritance, and what we know about modern genetics. And of course, these ideas have been, they've been reworked and revised and modified based on all sorts of other discoveries and experiments that we've made in recent times. So if we look at a brief timeline, you have Darwin's book on the origin of species, which you can see is now over 150 years old. Pretty darn good idea. It has not been rejected. His idea about evolution via natural selection. You had Mendel's work in 1865 that actually was kind of lost, and it was rediscovered in the 1920s and 1930s, and the experiments were replicated. And then you have Watson and Crick in the 1950s, along with Rosalind Franklin, discovering the structure of DNA. And think about that. That's the birth of molecular biology. Mendel was able to create formula, right, to explain the rates of change over time in generations of pea plants, but he didn't have this evidence microscopically, right? He was able to deduce that from phenotypes. When you get DNA, now we're looking at genotypes. We're looking at, different types of genetic code with A's and T's and C's and G's. So we're only like 70 years in molecular biology, in the field of molecular biology. So now what researchers do is we combine natural selection with genetics, and it seems quite obvious. We look at the production and redistribution of variation, and natural selection can act on this variation. Evolution defined. How do we define evolution? Well, evolution is simply just a change in gene or allele frequency over time. That's it. Now, we can track, look at genetics in both genotype and phenotype. That's what we learned, right? You learn the phenotype and genotype terms. You can actually track genes over time using genotypes over generations. You can actually track phenotypes because, right, the genotypes that certain organisms have dictate their phenotypes with a little bit of environmental interaction sometimes. So, if you have a lot of money and a big lab, you can track genotypes in a certain animal, like a specific lizard. Or if you don't have a lot of money and the background in molecular biology, you can track phenotypes like the color of the lizards. And scientists, we still do this. And so all you're looking at in terms of a definition for evolution, very simply, it's just a change in gene or allele frequencies over time. And you cannot prevent this from happening. It's incontrovertible. Every generation, the genotypes and phenotypes are going to be different. They might look the same sometimes, but their frequencies will have fluctuated. So evolution is a law, right? It's something that happens, and you cannot prevent it from happening. You'll see that the theory are the mechanisms, right? How do we describe this change? And that's what we'll get through in the next few lectures. But now we need to focus on, well, how do we measure this stuff? Well, we have to use population samples. You look at genotypes and phenotypes you've recorded in a sample of populations. And a population is a group of interbreeding individuals. Interbreeding individuals means you're talking about likely the lowest biological unit, which is a species. That these two individuals or people, everybody in a group, can interbreed. Think about the entire human species, right? Over what we're talking about, over 7 billion on the planet. Each person can interbreed with any other human on the planet, right? That defines us as a species. If we're going to study humans evolutionarily, then we're going to, grab a sample from a population in probably one part of the world. If we can find more and sample from more populations, then they would be more representative. That's what we would want. But the key here is that evolution evolves at the population level. You can't, you'll see many arguments against evolution that will be something like, oh, well, why don't you just go ahead and grow another arm? Well, that's not how evolution works. You have changing frequencies over time, and it doesn't, individuals are agents, but we have to measure this with groups of people. So here's an example of evolution. So the ABO blood group is, a good example. The blood types that we're already kind of learning about, right? You can be A, B, O, or you can be AB because of codominance. And we have a good idea that the ABO blood group follows Mendelian inheritance. What we can do is we can get a population of humans, for instance, and we can sample everybody's blood and find what your blood type is. We can look at your DNA and find out. And we can just talk about the alleles. We don't have to talk about genotypes, straight up alleles, right? Everybody has a genotype. Everybody would have 2 alleles per person, right? You'd have one on each pair of chromosomes. One you got from mom, one you got from dad. But let's just say we're going to sample everybody. Everybody has 2, right? Right, because traits occur in pairs, right? That was one of the rules that Mendel came up with. When it is one point on a gene that's responsible for one physical trait. So just understand that there are 2 of these alleles per person, but we're just talking about the percentage or the frequencies of the alleles when we first sample them. And we would, oftentimes you label that time 1 or here generation 1. So if you look at the frequency in generation 1, 50% of the individuals sampled have the A allele. 40% have the B allele, and only 10% have the O allele. Remember, the O is recessive. You need to have 2 copies to express the recessive trait. Generation 2, right? Let's say we have our population, and here's the trouble with humans, is that it takes a while for us to make a generation, because in humans, we have usually prolonged maturation. It takes a long time for us to be sexually mature. And this happens when a woman starts beginning her menstrual cycle, which typically can happen as young as even 11, 12, 13 years. So the interbirth interval in humans is like 13 years. But if we have a bunch of human adults, we sampled their DNA, we've got their genotypes and the allele frequencies. Let's trap generation 1 in a house and give them pizza and all sorts of food, and they're going to make a generation, right? I don't need to explain how this happens, but we have a controlled environment. They're going to make another generation, and then that generation, their offspring, we're going to sample their blood too. And we're going to figure out what the frequencies are of the ABO blood group alleles. And we sample that next generation from generation 1, the parents, right? Generation 2 is the offspring. And we see that now there is 30% of A, 40% of B, and 30% of O. The population has evolved. It has changed, right? You have more of the recessive alleles in generation 2, and you have less of the dominant A allele. All we're doing is describing the information and the evidence or the data. Didn't say why or how, which is the interpretation or the inference. And those are the theoretical statements. Those are the mechanisms. And we're going to focus on a number of them as we progress. You should all be aware that there are different scales to measure evolution. What's really challenging is, if you think about it, evolution is looking at generation time, and once we get into things like the fossil record and organisms that are extinct, we're actually using geologic time because we're looking at layers in the earth as a proxy for generation time. And there are different scales of evolution depending on how large of a swath of geologic time or evolution or generation time that you're looking at. So microevolutionary changes usually represent a few changes over, a generation or two, or maybe a little more. And then, of course, it all depends, what you're comparing it to. Microevolution is very easy to demonstrate in a laboratory setting. And you can do this typically with organisms that have a very short generation time, like bacteria, for instance. Bacteria are great to study in the lab and actually watch evolution take place because they can reproduce themselves quite quickly. You'll see a lot of research done with fruit flies because you can make another generation of fruit flies within a few days, and they only have 4 chromosomes, which makes it a little easier to manage. So those are models where we can actually introduce, and they have, for example, with fruit flies, where they've introduced some type of gene, right, into their genome that makes them more resilient to maybe some type of pesticide or spray. And they spray it at a number of different mosquitoes or fruit flies, and they die. And then the ones that were modified survive, and they only reproduce. And now you have a generation of fruit flies that are effectively all immune because now they all have that gene you inserted in them. So we can actually observe microevolutionary changes in laboratory setting very easily, and we can demonstrate it even in the wild with experiments. Macroevolutionary changes are basically the logical extension of microevolutionary changes, but now we're talking about larger swaths of time and generations. So macroevolutionary changes would be talking about, what we're going to discuss in the last part of this course, the transition from apes to us in the fossil record. And that's talking about millions of years of time, right? And we might block off a section as we're discussing to discuss, a few 100,000 years of time and changes that occur. So you can see that That's still macroevolutionary, but instead of hundreds of thousands of years, we might be talking millions of years now. So the two are the same, but we just have different scales in which to interpret them. Let's give some examples. So microevolutionary processes, a few classic examples, and these are courtesy of website through California, Berkeley. Look at the house sparrow size for males across the United States here on a map. We have red is the smallest body size for males, and blue, over to the blue in terms of the color, color scheme or hues, is large body size. The largest body size you can see are kind of in the northern latitudes of places like Minnesota and the Dakotas and all along the Midwest. And this might make sense because we typically notice that animals that live in more northerly latitudes often have larger body sizes so that they can retain heat. Usually also have shorter limb length so that they're closer to their body, which is where much of our central heat is, right? So that might be one way to explain, right, the distribution of male house sparrow size. But then you notice that the smallest is around kind of like San Francisco Bay Area. We might suspect the smallest ones to kind of be out here, out in the desert. And there are certainly, there's a trend like this on this chart. Now, we might not be able to explain this through climate. We might have to find other microevolutionary processes to explain this distribution. Global warming effects. If you look at the life cycle of the mosquito has actually been able to extend its life cycle so that it can travel to new, oftentimes northerly latitudes that it hasn't been able to habitate for who knows how long. What's going on because of climate change is mosquitoes are allowed to move further north. If you think about Arizona, we're going to be a perfect example of changes through climate in the introduction of new breeds of mosquitoes, which can also bring a whole suite of diseases. So we know that organisms are able to even modify their life cycle evolutionarily as long as it's an advantage to that organism. XDR tuberculosis, this is drug-resistant tuberculosis. If you've ever taken antibiotics, the doctor is going to tell you that you need to take all of it and around the same time of day, periodically, and don't miss a dose, as well as you're not supposed to be drinking alcohol because that will lessen the effects. So if you lessen the effects of an antibiotic, you don't take it properly, what you're basically doing is you're basically asking the organism to evolve and be resistant to it. And so we have a number of, diseases that plague us that have already evolved to be resistant to our antibiotics. If you are resistant to antibiotics after you get tuberculosis, I'm not even kidding you. You are going to be quarantined in a padded room somewhere in a research facility in Colorado because nobody wants another pandemic or epidemic of tuberculosis. You can see that some of the most dangerous diseases are the ones that are airborne and you can ingest, and tuberculosis is one of them. So we have all sorts of diseases that have evolved and become resistant to a number of our antibiotics, and that's a perfect example of microevolutionary changes. Macroevolutionary changes are sometimes much more subtle. Now, it's sometimes difficult to look at two organisms that are really different that share an evolutionary history. For example, when we looked at some of the DNA of elephants across Africa, there was one organism known as a hyrax that looked very closely related to it genetically. And a hyrax is a small, kind of furry mammal. And that didn't make sense at all. But when you think about, so many generation times that separate those organisms, that's why they look so different, right? Another good example are domesticated dogs. We think that domesticated dogs, that they evolved from organisms like foxes and wolves. Now, they don't look exactly like domesticated dogs, don't look exactly like modern foxes or modern wolves, but they do share some characteristics that you can obviously see that link them, right? So sometimes it's not always easy to talk about macroevolutionary changes when the two organisms or multiple organisms you're comparing are very different. Ones that are similar, now you have a way to kind of piecemeal together a long evolutionary history, and there's, again, only a few subtle changes. So if you look at coelacanth fish and other fish evolution, many of the fossil coelacanth fish here, they look exactly the same as modern ones. Matter of fact, you may see in the news, they did just, recover a very large coelacanth fish, right? They're hundreds of millions of years old. Trilobites. We think trilobites are the ancestor to beetles and pillbugs, which would be the descendants. Trilobites, they look very similar to modern beetles, and we can track their changes even in fossils as well as modern organisms, like traits like their like the number of ribs. You can see there's a timeline here. One great example of macroevolutionary changes are sharks, right? Sharks are a great example because they're hundreds of millions of years old, and in terms of their whole evolutionary history, the length of time they've been on this planet, and they look almost exactly the same, just a slight change in body size. Sharks in the past were much, much larger than modern sharks. But you can see that everything else, the fins, the teeth, the mechanics, are all the same.